All right, so let's start. This is the data set that has been sent to you. You will see that there are 4 files that end with the extension AB1, 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 AB1. So these are the actual chromatograms that are actually generated by the automated DNA sequencer. And you can open them in Finch TV, which is this software, or you can also open them in another package which we have called Sequence Scanner. Now, Sequence Scanner will create problems in installation, especially in cases where your Windows system is 64-bit. We do have a way of installing it. So what we have done is, since this is provided by the manufacturer of the sequencer, it allows you to look at many more details compared to the Finch TV program, which is, kind of limited in its view. So what I'm going to do is I'm going to show them side by side. So if I import a trace, now as I told you, the sequence files which are generated by the system are AB1 files, which are also called as sequence traces or chromatograms. So what you do is you locate your folder where your data is. And in your data, as you see, I have these 4 traces. So what I've done is I've basically opened import traces. I have browsed to the data which I have shared with you. So somebody is asking where to get those files, sir. There is a link in the chat box. In our Zoom chat box, there is a link which has been shared with everybody. You can just go to the link and download the data from there and save it in a more user-friendly location. You can open these AB1 files. You can add selected traces. Okay, so all of these traces are added, and now I click OK. So when I click OK, these all get added into my, so all of these got added into my sequence scanner package. And this is where Finch TV is on. In Finch TV, so in Finch TV, you do the same thing. You go to File, Open, or you can use this Open command, and you can locate where your folder is. So I have this folder called Data on my computer, and I'm going to open one of the files here. Now the thing is, in Finch TV, you can only open one file at a time. All right. And in sequence scanner, you double click on the file and you are able to see it below as a separate window, and you can open all 4 together simultaneously. Now, because Finch TV is a common platform for Mac as well as Windows, I'm going to show you the functionality of Finch TV so that it becomes easier for everybody to follow through. And we will minimize this other window. All right. So as you see, once you open the Finch TV program and you have this electropherogram opened or the chromatogram opened for you, you can scan through it through the entire length. And you can see that, the sequence is kind of okay with a slight bit of background noise. Here at around 70 position, you will see that there are multiple peaks. This typically happens at the 70th position and at around 120 position because of the presence of the dye blobs. The purification technology you use for cleaning up the sequencing reaction will decide how your sequencing output will come. So typically at the position 70 and at around 120, you will notice that if you use an older cleaning method, you will see dye blobs of unused or unclean dyes that are co-extracting within the sequencing reaction, and that's why you will have this, sort of a bulgy peak here. This is typically background noise because we know in the sequencing chemistry there is an inherent problem. But if you use enzymatic digestion or any other cleaning protocol, these dye blobs will not come and your sequencing platform will give you a clean sequencing output. All right. So you can have this, multiple views. If you use this view, you will get this. If you click on this wrapped view, your sequence will basically, entire sequence will be shown in a single field. You can increase the height of the vertical scale. As you can see, I can increase the height of the vertical scale by moving this bar on the left side. I can also move the horizontal spacing of the peaks by moving this horizontal scale. So this horizontal scale is there, and you can, have a view which is comfortable to your eyes. I wouldn't recommend increasing the vertical scale to a point where your peaks are saturated. So have a view which does not saturate the peaks. If you have this Q score, you see that the bars at the top, for those that are not able to view it in Finch TV, you can use the sequence scanner program to view the files. They should be opening there. All right. So this gives you the information. These tabs will allow you to reverse complement the entire chromatogram. As you can see, once you click on it, this particular option, it completely reverse orients or reverse complements the entire sequence chromatogram. So if you have a reverse primer that you want to check where it typically aligns, you can use this reverse complement option and correct for errors. There is this information, chromatogram info. So if you click on chromatogram info, you actually get the entire sequence, which is the FASTA file of this entire sample. You also get the information about the chromatogram, the sample ID, the sample name, which was given at the time of sequencing, the instrument model on which it was run, the instrument name on which it was run, what was the run start time, what was the run stop time, the lanes in the lane number in which it was run, the average base spacing between the peaks, so 12.58 scans. So that means the spacing between these two corresponding scans is around 12.58. This is the number of bases that have been called. So the total number of adenine calls is 235. Total number of sorry, this is this for C it is 446. For guanine it is 254. For thymine it is 391. All right. So this is what has been called for the average strength. The mobility or the base caller program that has been used by the machine is also typically given. This is called the KB base caller for the POP7. That is the polymer that has been used in the machine. The matrix and any other comments that the user has put in while they were doing this. These are the other details of the sequencing run. So there are many details which are available here in the program. So all of these are machine information as well as the current information as well as the starting scan point or the ending scan point all of these information is useful, especially when you try to figure out whether your sequencing reaction has worked properly or it has failed somewhere. So all of this information will be used to correctly check the accuracy of the sequencing run. So to edit the sequence electrophotogram, as I told you, you see this blue dashed line. This is the blue dashed line for QV20 or the quality value of 20. So when you see this blue dashed line and anything which is lower, let me change it to this. You see this blue dash line? Anything lower than this, I would not use. So my first, as you can see, the base spacing between different base calls is kind of even out, but the calling here is very disrupted. So what I would do is I would just select the particular base where I do not want to use. So what I would do is typically I would delete the first 20-25 bases which I'm not exactly sure of. So all you have to do is select the base and press the delete key on your keyboard. And that way you can delete the the bases that have been called by the system. So what we are doing now is we are manually curating each chromatogram that we have received from the sequencer and keep walking through. As you can see here, G, A, G. This is a correct call. So we go forward. So we have to keep checking through the length of the electrophotogram that our base calls are accurate. Oops. Now I will draw your attention to this point. As you can see, this is G, T, C, but there is a C and a T peak as well. So what I would do is I would mark this position. If you go to, IUPAC, if you put in Google IUPAC nucleotide codes, you can get this, and we will keep this open. But as you can see, there is a C as well as a T at this position. And if you go to that website, a C or a T is denoted by a Y. All right. So you can put a Y here instead of the C. All right. Now, because this is a clear non noise, it is not a background noise, but it is a clear peak because of which the signal is appearing. So you will put a Y here. And as you keep going along, in this case, the base strength is much higher compared to this particular one. So we will leave it the way it is. Here again, you see another base call, which is basically coming because of the blue signal and the black signal. So the blue signal is for cytosine and the black signal is for guanine. So you can correct this particular position again by looking at the C or G, which is S, and you can correct it for S here. So anything that you correct will be indicated in bold in this program so that you are able to visualize that correction at a later stage. All right. So if you keep scanning through, you see that your program has basically, again, as you can see, the peaks here are mixed. At this particular position, this position, there is a higher background noise. So I'm not going to use the data which is here. So I'm going to select this and press delete on my computer keyboard. So I'm deleting all these positions which are not being used here. So that way I can delete and only use the positions which are correct. So now this particular chromatogram is corrected. And what I'm going to do is I'm going to just save the chromatogram with the extension save as. So I'll go back again. I go to file and click save as, and I click on copy. So it is all automatically creating a copy of my original electropherogram, and it is showing it as a copy, and I save it. All right. So now I'm working on the copy file. So that way you can open the other chromatogram and correct it again. All right. So this is the horizontal scale. Now we are working on the organism 1704F. Remember, in the sequence name, you will also see that 704F is the primer name. All right. So this is the name which is typically given by the user when they are actually working on the files. All right. So now again, we go at this particular position because the base positions are very near, and the number of, the quality value is also very poor here. So we are going to delete each of these bases that have been called for by the machine at the start of the sequence. All right. So we have all of these corrections done, and we start from this particular T where our base calling is more rigid. Let's just confirm that every base has been called for at the start of the sequence. You will see that at the beginning of the sequence, the spacing between peaks is not really very accurate. And that is a common behavior if the polymer used in the machine is not a good quality polymer. So each position has been called for accurately. So we move across the length of the chromatogram file and see if there are any errors that are appearing in this particular chromatogram file. Now, this particular chromatograph file is from 704F. That means it is the forward primer. All right. Yeah. At this position, see, now at position 507, at position 507, again, you have two separate peaks. So one of the peaks is because of the G and the other one is called because of A. But the program has called it as an A alone. So instead of A and G, it has been put as an A. What you do is again, go back to this. A and G can be denoted by R. All right. So what we are going to do is we are going to place an R here. All right. So we have selected the base and we have, just replaced it with an R on our keyboard. So go ahead again, keep going, and keep watching this blue line and these bars. If this blue line, if the bars are shorter than the blue line, you need to make sure that the base call is accurate. All right. So now, once again, you need to make sure that there is a C here, there is a C here, there is a G, C, 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 G, T, and C. That's it. There is no other call. So this sequence is now checked and corrected. All right. So we will again file, save it as a copy, and we save. All right. So we can do that. Now we'll go back to the uh, third file. So before I tell you, um, I told you during the theoretical part that when we sequence the 16S, we usually sequence it using internal primers. And as you can see, the sequence is generated using 518R, 704R, 907R, and 1028R. So we have used 3 reverse primers and 1 forward primer to get the entire sequence. So now we check the correctness of 907R. So we select this particular sequence, open it in our Finch package. We, change the view to our comfort level. All right. And then we correct the errors. So as you can see, the base calls are correct here, but the base spacing is more here. So what I'm going to do is I'm going to start deleting from here till the backside. So this is deleted. So we are deleting all of these because those bases are not accurately called. They have a quality value which is lower than 20, indicated by the blue line at the top. All right. Now all of these bases are okay. So let us move ahead a little. Now, as you can see, once we deleted the initial bases, the numbers have shifted. So now this has become 10, which was originally probably around 30. Okay. So this will change the shift. And as you can see, when I told you that we usually see diblobs at around 70, now they should be, nearing around 50. So that is the difference that has been caused. Now we scan through the chromatogram, we do not take care of the background noise, but anywhere, wherever there is a double peak, we have to correct for it. We have to correct for ambiguities. So we will go. If you have questions, please raise your hand so that I see that a question is coming. It is easier for me to track the hands raised rather than the questions which are typed in the Q&A box. Okay. So as you can see, we have scanned through roughly, 500 bases. We have not seen any, so-called SNPs as we saw in the other two chromatograms. We are still okay. Here, this is a base I would like to draw your attention. Even though the base call is low, there is a clear peak here. This is a decreasing signal from the previous A. So we will not consider this as a peak since it is on the shoulder, not on the peak. Now here again, we have a G by A. Now, oops, sorry. We have a G by A, which is basically a background noise. But here, this is a clear peak. All right. So we have to correct for this C by T. So what is C by T again? C by T is Y. That means it's a pyrimidine base. So we will correct it for Y. We will scan through the remaining part of the chromatogram. Now, there is something I would like to point out here. As you see, the background noise has increased, and the peaks are not so clearly separated out at this point. All right. So this clearly indicates that the signal is no more good at this place. All right. So we will try to terminate this particular signal at this position where we are not able to correctly separate out the noise from the signal. All right. So we have to start terminating this signal or deleting the portion ahead of this sequence up until the end, which is roughly about 880 bases. And we have to do it starting from, about this position, where we see that multiple base calls are below the Q20 value. So Natasha Chakrabarti, you have a question. Please go ahead. Unmute yourself.
So whatever sequence which comes in between the like whatever nucleotide which comes in between the sequence has a QV below 20, considering that there are no double peaks in that nucleotide. Like what kind of change do we make to it? Like do we delete that or do we make any kind of change to that?
So if I'm not wrong, what you are suggesting is on the computer, when I say that this peak is G and this peak is C, but there is a nucleotide base between the two. Is that what you are suggesting?
No, sir. What I meant is that like if the G at 630 position is
Hmm.
below the blue line, like below the blue line is QV below 20, right?
Hmm.
So what kind of change do we make to it
No,
since
we
it
don't
comes
do
in?
any change. We don't do any change unless there are multiple peaks below that G.
Okay, that was my
Okay?
question. Thank
Yeah.
you, sir.
Yeah. Okay. You can lower your hand. Um, Baguma. Baguma, go ahead with your question. Baguma, you can
No, no,
unmute and
please,
speak.
no, please, please. That is
Yeah.
a problem of my matching of my laptop. Please.
Okay. All right. Okay. Yeah. Thank you. Lower your hand, please. Yeah. All right. Siva, Dr. Sivakumar Kannan, any software to automate the process and curation of DNA chromatograms? Yes, there are many softwares. In fact, the software that we are going to use next can also do this manual job automatically. But then the problem is we don't really use that often, especially when we are handling single sequences and the sequences are supposed to be very, very crucial for our data set. Chaudhry Saddam Hussain, what is the reason behind this to remove these bases from the sequence? Well, it is quite obvious that the reason behind removing sequences or bases which are not following the pattern between two peaks, they have likely been called by the software because of an error. And corrections of peaks we are doing because we believe that the software was not able to identify or separate the background noise from the actual peak, and that is why we are doing doing these corrections. All right. Yeah. So let me read one more question. Okay, there are no more questions. So now the problem with Finch TV, as I told you, is you have to select individual base and delete, right? Whereas in the Chromas Pro package, you can do this like you can just select the entire portion and delete here also. All right. So you can just select the entire portion till the end and delete the entire portion, which is erratical after a certain point. So let me correct here. Let me remind everybody that once you delete bases, multiple bases from a sequence, because the sequence after that is not accurate, you cannot just selectively delete portions uh, from within the sequence and keep portions at the end of the sequence, because then your entire sequence will change. So if you are deleting certain portion from the end of the sequence, please make sure that you do not keep any of the portions in between. Otherwise, you are going to create a complete mess of your data. You have to keep the data in the middle. You can only delete portions at the start or at the end of the sequence, which are completely erratical and cannot be determined by the machine. All right. And that's why we have deleted the entire portion. We have not kept, like I have not done that. I have deleted these, 10 positions and kept these 5 positions, again deleted these 10 positions and kept these 5 positions. I am not doing that. Once I am sure that the data is not accurate after a certain position, I am going to delete that entire data till the end of it, because otherwise you are going to create artificial positions. All right. So again, we go back and we click Save As, and we have done Save As for this. Now we open the last sequence, which is 1028R, and we also correct this portion. We delete the starting portion, which is not accurate. We keep the middle portion. This is the die blob. So the base call is still accurate. A, and a G here. This is G, A. So this is accurate. Because of the die blob, we have to ensure that position number 70 and 120 are accurately called. We go ahead. The sequences are clean. There are no double peaks. We do that and we see that. Let's see here. Now again here, as we see, the position between AA is, the spacing between these two A's is less, but the spacing between the A and C is more. All right. So we consider this to be accurately called. We move ahead. Now look at this G. This G has no QV score above it. So that means the machine is not able to accurately determine anything after this. All right. And as we can see, we have started coming across rolling hills or peaks that look like background noise, but may or may not be actual peaks. We have these rolling hills. We have, for the same distance, the machine has called 2 Gs, but for the same distance between these T, the machine has called 3 Ts, and we are not sure what is happening here. So we believe that the data is not accurate after this G call. So I'm going to delete the entire data until here. All right. Until the end of it. So we have now got enough good quality data. We again save this as a copy and we keep it. Okay. Now our work for manual curation is done. We have used Finch TV to accurately correct for errors in the chromatograms which were used by the machine, which were delivered by the machine. Now what we do is we go back to our software tools. The next software tool that we are going to use, as given in the email and the chat box, is that we now need to build the context. So for that, we are going to use Chromas Pro, which we have already started here. What we do now in Chromas Pro is we open Chromas Pro, we go to File, we go to New and create a new sequencing project. All right. We create a new sequencing project. And in the new sequencing project, we now go to project and add files. These are the files that we have now created as copies. So we now will select copy, 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 copy. You can select these four simultaneously by pressing the control key on your keyboard and select all four and click open. Now they have all opened and got imported into the software, into the Chromas Pro software. All right. So these four files are there. Each of these are available now. And what I'm going to do is I'm going to assemble all. So what it does is, as an algorithm, the computer program basically creates a config out of these four files. So this is a config which is having a length of 1404 bases. Now, if I open the config, I double click to open it. I maximize this window. And if you move the cursor along, you will see that my 704 sequence is actually being used as the first sequence. And as I travel along, my other sequences start getting added. All right. As you can see, my deleted portion is also incorporated here, but without any base calls. All right. So this is how one below the other, the exact position overlaps are being, calculated by the software, and it is giving us a full length sequence of the sample. All right. It has given us a full length sequence of the sample, which is roughly 1404 bases long. Now, there is one thing that I would like to point out here is that my 518R, 907R, and 1028R have been put in the reverse orientation, whereas my forward primer should be the one that should come later on. All right. If you look at our presentation where we had shown these sequences, when you determine the sequence using 27F primer, it is determined in the forward direction. But when you use a reverse primer, it is used in the reverse direction. It is determined in the reverse direction. So when you are actually building the contigs, for building the contigs, you must reverse complement the 907R so that it comes at the front. 518R reverse complement, so it comes at the front. So in Chromas Pro, what we are going to do is we are going to go to reverse complement, go to edit and create a reverse complement. All right. So if you create a reverse complement, you will see that now your 518R, 907R, and 1028R are the beginning sequences, and the forward primer is the ending sequence. All right. So that is how the config has been made in the right orientation. All right. It is made in the right orientation. And now what we do is we go to File and say Export to Editor. When you export to editor, your entire full length sequence is being saved as a single file. And the full length sequence from the window, this one, is the top sequence which is coming at the top here. All right. It is the consensus sequence as we call it. The consensus sequence is generated from all the electropherograms together. All right. This is the consensus sequence which is starting from GTTT. And if you go to your text file, it is starting from GTTT. All right. So that clearly shows that it is the entire full length sequence which has been copied. So save this in the same folder. You can save this as a sequence file, or you can save this as a FASTA file. So if you save this as a FASTA file, it'll be easier. So you save this as a FASTA file with the name organism1. I have already saved this file for you, so you can check. All right. So now we close this window and you can save this project. You can save this entire work that you have done as a project. You can save it as organism1 GPJ as a project file, and you should be able to come back to it later on whenever you want to use it. All right. So we have a question from Zikrul. Zikrul, you can unmute yourself and ask the question.
Yes, sir. Should we push the reverse button before saving the file on what is it? The Finch TV, or after we put into the Chromas Pro?
If you incorporate anything in Chromas Pro, it will automatically, using the algorithm in the system, it will try to align it in both the orientations, and whichever orientation fits better, it will select that. So whether you reverse complement before incorporating in Chromas Pro, it will not matter.
Okay, okay.
Because
Thank you.
Chromas Pro is based on an automated algorithm, it will automatically try to adjust based on which orientation fits well for which primer. So it tries to identify overlapping regions and then builds a context for you for all the sequences. My idea of telling this to do afterwards is because the reverse complementing then will happen for the entire sequence. So that will allow it to be used better. All right. So now we have saved our sequence, our full length sequence. If you go back to our folder and look at the FASTA file, you will see that the FASTA file is typically, saved like this. So it has this, greater than sign followed by the name of the sequence. You can change this name to organism 1. Okay. Instead of context 0, you can save it as organism 1. And this is the FASTA file that is saved. Now, what we are going to do is we are going to use this FASTA file to do the blast analysis or easy biocloud analysis. So what I'll do is I'll go to internet and type NCBI blast. This will come as the first hit. You can go to NCBI blast and go to nucleotide blast. Okay. In nucleotide blast, you can choose the file directly from here. So this was the file organism1 FASTA, or you can also copy paste the sequence into this window. Now, when you do this analysis, what you need to do is you need to check against the ribosomal RNA and ITS database. This is because you have used 16S rRNA as your target gene. All right. So you select the 16S ribosomal RNA, bacteria and archaea database. And I also suggest that you use sequences from type material. You check this box and use sequences from type material. So what it will do is it will allow NCBI to restrict the search only against organisms which are described as type species under the ICNP. All right. And use highly similar sequences and click on blast. When you click on blast, you will see, you will have to wait for some time until the window opens up. It is just taking a little bit longer than I thought. Should have been pretty straightforward. What we can also do now is we can, in parallel, open our Easy BioCloud. So you can go to you can type Easy BioCloud. Okay. And when you go to Easy BioCloud, it will give you this page. Easy BioCloud allows you to create, to use the website as an academician without any cost to it. You don't have to buy a license, but if you are a commercial entity or a private entity, you will be required to buy a license. So you can go to Easy BioCloud, create a login ID and log in onto the page. My IDs are already fed into the system. So I have logged in now. And what I do is I can do the same thing by going at 16S based ID in Easy BioCloud. And what I can do is I click on this identify new sequence, and I have to go back to my FASTA sequence. I copy this sequence from here and paste it in this window and type the name of the sequence, which is organism 1. Okay. And I click next. And it says sequences loaded 1, organism 1, base pair 1404. And I click submit. On this page also, it will take some time. And on this page, our results are already here. Okay. So as you can see, if you look at a graphic summary, you will see that certain sequences which have matched our sequence are shorter than the sequence that we have used. Our sequence is 1404 bases, whereas this sequence is shorter, and that's why it is showing this much region, whereas most of the sequences are nearly aligning the entire region that we have checked. As you can see, alignment scores, if these bars are in red, that means over 200 bases are aligning for our sequence. All right, this is the score. So, and if you want to look at the actual alignments, you can go to the alignments tab and you see that the first hit which NCBI has given shows 1,399 identities compared to 1,404 bases. So that means there are 5 mismatches. Out of those 5 mismatches, none of them is a gap. So let us see where are those 5 mismatches. This is a mismatch. This is a mismatch. Let us see. This is a mismatch. This is a mismatch. All right. This is the second sequence that it is matching to. So that way we have, a lot of mismatches. If you look at the taxonomy, what it is showing is our organism belongs to Bacillus family, the Bacillus genus, and from the Bacillus genus, it belongs to the Bacillus subtilis group. And within the Bacillus subtilis group, it has shown, matches with so many organisms. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15 different species within Bacillus subtilis group. All right. And if you look at other Bacillus species, it is Nakamura and all of these other things. Now, if you go back to the first page, which is descriptions, you see that the query coverage, the e-value, the percent score is way different for all our sequences. All right. So we have this that we are checking for. Now, as you see, this particular tree output has been sorted based on the e-value score. Whereas if you go to percentage, if you click on percentage identity, it will sort it based on percentage identity, where it is showing the hit with Calidae Fonti Bacillus Erzurumensis, strain P2. All right. The second highest similarity is with Bacillus amyloliquefaciens with 99.78% similarity. Again, these strains, 99.71 is amyloliquefaciens again, 99.64 is Bacillus subtilis, 99.57 is Siamensis, Vallis, Mortis, Nakamura. So we have all of these different strains that we have checked. But as you see, the query coverage with the top hit is only about 96%, 98% here, 98% here. So there are a lot of differences that we see. Okay. So based on all of these differences, we will have to see what we want to select as the closest homologue. All right. So if we sort it based on query coverage, we see that Bacillus subtilis, Siamensis, Nakamura, Bacillus subtilis, stercosis. So all of these organisms come. So I typically leave it to the default. And what I typically do is I would select multiple species which are unique that come out of from my top 100 hits within the database. One can also download the entire database and you can select columns. Okay, you can select all or you can select whatever you want to download as your sequence. So I've already selected Subtilis, Siamensis, Vallis Mortis, Nakamura, Stercotis, Cabrialis, Amyloliquefaciens, Iniquosorum, Rugosus, Nematocida, Halotolerance, Mohavensis, Vallis Mortis is already selected, Tiquilensis, Spizizani, Atrophius, Mohavensis, and let's also identify this particular one. Okay, so now we have selected all of these. What we can simply do is we can go to FASTA and we can go to complete sequence. We can go to align sequence or text. All right. So whatever you want to download, you can download. We will select FASTA complete sequence. And our file is downloaded. We open it up. And as we see, our sequence is probably empty. So we have all of these other sequences that we have downloaded from NCBI. All right. And we have our original organism is not here in this list. All right. So we have this organism one already opened out. So what we will do is we will copy this and we will paste it at the top of our data set or the sequences that we have downloaded. So now we have all the sequences in a single file, right? So we have all the sequences in a single file and we are going to just save this as sequdump.txt. This is how the name has been given, but let's just save it as, let's go to all files and go to our actual folder and save it as organism1.fasta. I have selected all files instead of text document in the type, and I have put an extension .fasta and full name organism1. So that gives us a new file organism1.fasta, which is now available with us. Okay. So now in order to build the phylogenetic tree, we have to align all of these sequences. And once we align all of these sequences, we should be able to build a phylogenetic tree here. All right. So Now we open Mega package that we have already sent the link to, and you guys have already installed the Mega program in your computer system. Yes, Zikrul.
So I would like to ask what happened to the file you uploaded into the EasyBio Cloud?
Okay, right. Yeah. So EasyBio Cloud has also given us an output, and we see that organism 1 is identified as Bacillus siamensis, strain number so and so with this much identity. All right. So if you click on this amplify button, we see this output that has come up here, 1404 bases, and the orientation in which it is matching is forward. And in EasyBio Cloud, the 16S database and the algorithm is actually much more, what should I say, evolutionarily robust. So it automatically gives you the top hit is almost always the closest hit. So if you are using EasyBio Cloud, I don't recommend doing additional phylogenetic analysis simply because EasyBio Cloud has a built-in feature where it basically, starts the where it basically calculates the dissimilarity between sequences and does an evolutionary assessment of the top hit. So the top hit in EasyBio Cloud is almost always your closest hit. And if you read the literature about EasyBio Cloud also, that will tell you that. So in this case, it is already identified as Bacillus siamensis. If you click on this name Bacillus siamensis, it tells you the number of genomes that are available, the number of 16S sequences that are available. If you go back and click on this view alignment, it shows you the entire alignment that has with which your sequence and the hit sequence, that means the sequence which is in the database, which is Bacillus siamensis, how well aligned they both are. All right. So there is one base difference here. There is one base difference here. Now this is R. So that means it could be a G or a C. So this is still okay. As I see, only these two base variations occur in this particular sequence. So because of which your sequence is pretty much your organism is now identified as organism 1 in this case. All right. So this is what the output of your EasyBio Cloud will look like. Now my Mega has opened up, and so what I'll do is in Mega, what you can do is you go to File and say open a file or a session, and go to your location of the file where we have saved that organism1.fasta and click on open. Now it asks you whether you want to analyze the data or align the file. Now let's say we want to analyze the data. So we just click on analyze, and it asks you whether these are nucleotide sequences or protein sequences or pairwise distances. Our data is nucleotide sequences. So we click on OK. We select nucleotide sequences and click OK. And this is what the data looks like here. Now it has shown an error. So what we are going to do is we are going to check where that error is coming up. There doesn't seem to be any error on my side. Okay. So let's go to data is already aligned. We close this. We go to File, Open Session. Let's select that file again and click Align instead. Now, as you see, when you click on Align, all the sequences have been imported into the mega package. This is your organism 1, and all of the others are your other organisms of interest which you have chosen from the database of BLAST. You can simply go to alignment. You can select all the sequences here by pressing the Shift key and selecting all. Go to alignment and click Align by Cluster W. Okay. It gives you these parameters. I don't change these parameters. I keep them like this. And it does a pairwise alignment. Then it does a multiple alignment. Okay. Now, once the multiple alignment is done, as you see, before the alignment was not there, none of the bases were properly, of the same color. But now, as you can see, they are all of the same color, right? So they have been aligned perfectly using this algorithm, and the alignment is nicely done, very accurate. And all you have to do is now, there is one thing that I would like to point out here. Many researchers have this criteria of using only the same portion of the alignment. So whereas some others use a conservative approach of not losing the data, and what they do is they use enough data so that majority of your sequences start at the same point. All right. So in my case, let us say if I use this position, out of the 17 sequences, 3 plus 3, 6, 7, 8, and 9. Only 9 sequences would be starting from this position. If I use this position, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 positions or 12 sequences out of 17 sequences will start at the same position. If I use this position, I'm going to add 1 and 2 more sequences. So out of 17, 14 sequences will begin at the same position. So 14 sequences out of 17 sequences is roughly 82%. So that's, above 80% of the sequences are starting from the same position. So what I'm going to do is I'm going to delete the data ahead of this. So what I'm going to do is I'm going to select these columns. Okay. I'm going to select these columns and I'm going to press delete selected block. So what it will do is it will delete all these positions from the data set. However, doing this will also delete positions that may have information for building the phylogenetic tree that may provide robustness to the phylogenetic tree. All right. So it is your choice and your judgment that will decide what you want to do, whether you want to keep all the positions, all the sequences starting at the same position, or you want to keep majority of the sequences starting at the same position, or you want to keep all the sequences the way they are. So basically, there are researchers who want to use the entire data that is available at their disposal. There are people who want to use data which is conserved to 80%, and same thing people would do at the back end of the sequence. All right, at the terminal end of the sequence, people would try to, delete portions which are, not having any data for some of the positions or some of the sequences. So this is usually done when, you want to be very, very accurate and rigid. So I personally use a conservative approach wherein I try to use only majority of the data starting at the same position. So I would delete these starting blocks, and same way at the back end of the sequence, I would delete portions which are not present in majority of my sequences. So let us say if I delete positions from here till the end. Okay. So that is what I would do in my case. Now my alignment is done and I want to save this alignment and I want to do a phylogenetic analysis of this data. So first of all, to save this alignment, you can save export alignment in mega format, FASTA format, or Nexus format. You can choose whichever you want. Or you can directly go for phylogenetic analysis. It will ask you whether this is a protein coding nucleotide sequence data. Now, because this is 16S sequence, it is not protein coding. So this is no. All right. I'm going to do this minimization and go to phylogeny, and I say construct maximum likelihood, neighbor joining, UPGMA, whatever. So for ease of purpose, I'm going to show neighbor joining. Would you like to use the current active data? Say yes. Now this is where the trick comes in. Scope, you want to use all selected taxa. Statistical method is neighbor joining. Test of phylogeny can be chosen as interior branch test, bootstrap method, or none. We prefer to use bootstrap method, number of replications 1000, and you want to use a nucleotide substitution type. You can use any model, Jukes Cantor, Cumera 2. There are other models as well. You can use P distance. I prefer to use Cumera 2 because this is more robust. There is a manual of Mega that will help you understand the algorithms, why you choose a certain algorithm, why you don't choose a certain algorithm, and so on and so forth. Okay. So you can choose that. And I always prefer to use pairwise deletion instead of complete deletion as the gaps and missing data treatment. And I select OK. All right. So it is doing 1000 replications and it has given us this outcome of the tree. And this is the bootstrap consensus tree where my organism 1 is actually matching to Bacillus siamensis KCTC 13613. All right. And its second closest relative is Bacillus amyloliquefaciens. The third closest is Calidae fontibacillus, and the fourth one is Bacillus nematosida. Now, if we go back to our easy BioCloud results, you can see siamensis is followed by velezensis, followed by Bacillus subtilis, followed by amyloliquefaciens. Okay. If you go to the blast results, Bacillus subtilis was the top hit, siamensis was second, Vallis mortis was third, and so on and so forth. If you want to change it to percentage identity, it is completely opposite with Calidae fontibacillus as the closest, amyloliquefaciens as the second, subtilis as the third, and siamensis as the fourth. All right. So as you see, the results of your phylogenetic analysis and the results of your blast results vary drastically. They will differ many a times and in most cases. All right. So it is very, very important that you always perform phylogenetic analysis by selecting sequences from your data set that are close relatives of your organism. And with this, as you will notice, our organism has been determined to be Bacillus siamensis, closely related to Bacillus siamensis. All right. Anybody has any questions so far? If you want, you can type your questions in the chat box or, other things. We have questions.
There
Oh.
are some questions in the chat box already, Dr.
Yes,
Kamlesh.
yes. I just looked at them. So I'll stop sharing my screen and I'll answer questions first. So the first one is, according to my observations, Easy BioCloud is good for 16S sequence. Yes, it is. I always prefer to use Easy BioCloud. And being a culture collection, we always rely on the sequences that are present in Easy BioCloud than in NCBI. Dr. Sivakumar is asking, is Easy BioCloud limited to prokaryotic sequences or does it also encompass eukaryotic sequences? Frankly, I'm not sure. Let us see. It is a whole genome analysis, detection of SARS-CoV-2, genome-based 16S, shotgun-based metagenomic profiling. I have a feeling they are now expanding to other data, other organisms as well. So they are expanding to other organisms as well. The next one is from Zikrul. Sir, what about EasyBio? Yeah, so Zikrul, we already discussed that. Muhammad is asking, sir, how you got those 4 files that you opened in Fitch TV at the beginning? Okay, Finch TV, the data that was given to you as a link in the chat box, the data was downloaded from there. Okay, so you can check that. The next one is from Ravi Kiran Jadhav. The question is cluster W and muscle. Which one is correct method for alignment? Either of them is correct. Each one is based on a different algorithm. So depending on the number of sequences you have in your data set, I use cluster W when I have fewer sequences and I require more accurate alignment. I'm just more friendly with it. I would use muscle if I have too many sequences in my alignment to do. The question is, in this bootstrap values are below 60, then in that case, tree is accepted. So here is a judgment about phylogenetic trees. And the judgment about phylogenetic trees is, out of the 1000 replications that the tree has done, it has arrived at 50 times more occurrences than any other occurrence for this particular combination of clustering at this particular node. Okay, so for that reason, there is no acceptability or deniability of the tree. There is only robustness of a tree. So if you feel that there is no more accurate noding or clustering pattern compared to this one, and this has a better bootstrap value, this is your robust tree. Okay, so that is how I take it. Obviously, any value which is above 50 is considered to be better. But it is not that values below 50 are not accepted. If you talk of acceptance, that is a different scenario. But the robustness of a tree and a certain bit of, degree of uncertainty associated with it, yes, I agree that trees below 50 are used very cautiously. I hope I'm able to answer that. Yeah, so Kumar has asked, sir, what are the values left side in the phylogenetic tree and indicates what? So yes, Kumar, let me share the screen again. Yeah, so the values on the left, these values are bootstrap values that support the clustering pattern at this particular node. Okay, so if you look at this node, that means organism 1 and Bacillus siamensis at this particular node appeared 50% of the times. That means out of the 1000 replications, it appeared 500 times together like this. And if you consider the clustering pattern of this node, that means these three organisms coming together, they appeared 43% of the times together. All right. So this is what it means in this value. There are a lot of features in Easy BioCloud that one can use. You can actually there are different tree types, subtree, view, tree, branch style. Yeah. So you can also look at a radiation like this. You can also look at a circle like this. So Mega allows you to make trees the way you want. So you can choose the design that fits your requirement, even in traditional rectangular, straight, and you can also have a curved one, which is more curvy here. But I prefer to go with the traditional one that is there. You can give colors, you can compress nodes. So let's say you want to compress a node. There's a compress, expand. So if you compress this node, you click here and it gets compressed. You can click again, just cancel it, click again, and it'll expand again. So you can use this Mega software to build the trees and display trees the way you want. All right. Now, next question is, this was done. Sir, what if we get two different strains or organism using Mega software and Easy BioCloud? Which one would be preferred as selectable and why? So, I would always rely on my phylogenetic analysis first. But before I blindly rely on it, I would like to cross-check my data. So first check that you should do with Mega is that if your alignment is accurate, you should see these colors the way they are, perfectly aligned one below the other. Same color bars should be there. At times, if your alignment is not accurate, your phylogenetic tree will be problematic. So you should not use a bad alignment and conclude based on the phylogenetic tree. All right. So I would always rely on my phylogenetic analysis rather than relying on only Easy BioCloud or blast analysis output. All right. The next question is from, can you explain how pairwise deletion works in phylogenetic tree construction? Yes, absolutely. So how it works? So let's go at this particular sample. So when the computer algorithm is comparing organism 1 with organism 17 to calculate the distance between them, it is considering that these positions which do not have any data are also absent from my original organism. But when it is comparing organism 1 to organism 5, let's say, both the organisms have this data set with them. The positions have nucleotide bases. So when comparing organism 1 to organism 5, it will calculate the distance considering that this data is present. And for organism 1 versus 17, it will consider these positions to be absent from both the data sets. All right. So that is how pairwise deletion works. And that's the reason I use pairwise deletion whenever I'm using phylogenetic analysis. So Ravi Kiran has a humble request. Please make available recordings of this meeting for participants. Recordings will be available in 48 hours on our YouTube channel. So please subscribe to our YouTube channel and you should be able to see everything there. And I think, Aaron, there are no more questions. I'm done. Okay. So thank you, participants. Thank you, everybody. And thank you for patiently listening. We started with about 150 and we are still at 70 plus. So that's a good number. Yeah, Aaron, I'm done.
Thank you very much. So the next coming up one would be on, yep. So the next coming up session will be next month on the third Saturday with Professor Lorena Caro from University of Salamanca, Spain, on the analysis of genomes and microbiomes. So this is second part of the series. So we must thank Dr. Kamlesh for his very well thought out session with us and workshops. So if you do have any questions, you can email him and contact him. And of course, you can follow us on our YouTube for the repeats if you need to just revisit the sessions. There are many previous sessions. One of it is related to Easy BioCloud. So if you want to see that, that would be the year 2022, the 16th July session. So that is also on Easy BioCloud. So you can revisit that. So with all that said and done, thank you very much for attending today's BISMiS Live. I hope you have gained as much as I have. I'll see you again next month